we are very well prepared for looking at antigen antibody reactions. Just to give you a glimpse of our previous lecture, because we have looked at the antigen in the couple of lecture back, I will not go through the entire slide. It is just for your revision. So anything which is recognized by body as a foreign will be called as antigen, and you know they can be self and non-self. And we have also seen what was an epitope. Epitope is a part of an antigen, and it is also called as antigenic determinant. We have also seen it, so we'll go ahead. Now, let us talk about what do you mean by antigen antibody reactions. So basically, the antigens and the antibodies, they combine specifically with each other, and this interaction between them is called as antigen antibody reaction. So this reaction can be abbreviated as AG-AB reaction. AG stands for antigen, AB stands for antibody. And this reaction forms the basis of humoral immunity or antibody mediated immunity. So by now you very well know that because antibody is a part of humoral immunity, this reaction is also a part of humoral immunity. Now, when I talk about reactions, you should also know what is actually a reactant in this reaction. So basically, when antigen and antibody interaction occur, this interaction is basically occurring via binding of epitope and paratope. Now, epitope we have just seen, it is a part of pathogen, that is antigen. Antigen का part होता है epitope. So epitope of the antigen will react with the paratope of the antibody. Now we have not used the term paratope before. We have even seen the detailed structure of the antibody, but we have used some different term instead of a paratope. Now what is a paratope? If you are looking at the pointer where I'm pointing, this part we had called antigen binding site. So paratope is nothing but antigen binding site. We had in a previous lecture studied antigen binding site in detail. You know that this is a variable region of an antibody which consists of initial 110 amino acid. It has 3 CDR, right? Within this CDR, not all the amino acid binds to the antigen. We have seen that the CDR region is responsible for binding to the antigen. But I am saying not all the amino acid in CDR are responsible for binding to the antigen. What is the paratope then? The paratope are the amino acid sequence within CDR which are actually binding to the antigen. So your paratope is nothing but within CDR, which is nothing but at the antigen binding site, which is binding to the epitope of the antigen. So this is the epitope and it is binding to the paratope. And this is how your antigen antibody reaction occurs. I hope everybody is clear till here. Now when this reaction occurs, it occurs in 3 stages. And the reaction involved different types of steps. What are the 3 stages of reaction there? A primary stage. In the primary stage, there are no visible effects. When antigen binds to the antibody, there are no visible effects. It occurs very rapidly. It occurs even at a low temperature. Most importantly, it is reversible, and it involves weaker forces like van der Waals forces, ionic interaction, and hydrogen bonding. So you can see over here, it involves hydrogen bonding. ये antigen है, ये antibody है. It involves hydrogen bonding, ionic bonding, hydrophobic interaction, and van der Waals interaction. So everybody understood. So first stage in antigen antibody interaction is when epitope binding to the paratope. The interaction is not visible. It is reversible, and it involves and weaker interactions like van der Waals effect, ionic effect, hydrogen bonding. The secondary stage is when if paratope and epitope are complementary to each other, then this is going to lead to the demonstrable events like a precipitation, agglutination, and killing of antigen, which is also called as neutralization, and the neutralization of toxin, and fixation of complement, immobilization of motile organism, and so on. So you can actually see the effect. So these are the visible changes, the precipitation, agglutination, these are the visible effects that you can see in the secondary stage. And this happens if the paratope and epitope are complementary. Tertiary stage is inside the body in vivo. Secondary stage can be visible even in vitro. If you react in vitro your antigen and antibody, the secondary stage can be seen. Whereas tertiary stage is only seen inside the body. That is the killing of the antigen, which is called as neutralization, and the which eventually sometimes leads to the tissue damage. So these are the three stages, important stages of antigen antibody reaction: primary stage, secondary stage, tertiary stage. In a coming lecture, we will be more focusing ourselves on a secondary stage, because as I mentioned that the secondary stage, that is antigen antibody reaction, can be visually seen if you perform antigen antibody reaction in vitro, that is outside the body. In a test tube, if you react antigen and antibody, then you can see that reaction visually outside the body. So we are going to focus more on a secondary stage in coming lectures. Now, what are the features of antigen antibody reactions? The first feature is the reaction is specific. Of course, the antibody is basically targeted to antigen, so the reaction is most of the time very specific. But there can be an exception, because sometimes the epitope, that is a part of the antigen, is shared between multiple organisms. The multiple types of species, of the multiple types of organism share the similar epitope. If that happens, then a cross reactivity may occur. Second, the entire molecule react and not the fragment. Third, no denaturation of antigen or antibody occur. Fourth, combination occurs at the surface. That is, antigen antibody reaction which occurs at the surface is more relevant to us. Fifth, the reaction is firm but reversible. Reaction of antigen antibody which is occurring at the surface is firm, but that is reversible. And what is more important, the affinity and avidity of the reaction is more important. Now we'll see this term a little in detail, that is what is affinity and avidity. So basically the firmness, how firm the reaction between antigen and antibody is decided by affinity and avidity. Now what is the difference between affinity and avidity? Affinity is intensity of attraction or closeness of fit between epitope and paratope, whereas avidity is strength of a bond after the formation of antigen antibody complex. Now, this is more easy to understand with the example. Now, if you see, this is an antigen. Now, to the same antigen, IgG is binding and IgM is binding. Now, here, if you see closely, if you imagine, this is a lock and this is a key. Then, and this is also if you see epitope is a lock and a paratope is a key, then you can see the more fitting key is IgG than the IgM. Okay, so the affinity refers to the strength of a single antibody and antigen interaction. So you feel that the IgG is fitting more well into the antigen. Whereas what is happening when the IgM is binding, it may not be fitting that well as like a IgG, but the overall strength which this complex achieves after binding will be more than the IgG. This is because the number of a binding site with the IgM are more. So I'll read out now. Affinity refers to the strength of a single antibody antigen interaction. Each IgG antigen binding site typically has a high affinity for its target. It has a high affinity, a good fit to the target. Whereas avidity refers to the strength of all interaction combined. IgM typically has a low affinity antigen binding site. This does not fit good as compared to IgG. So it has a low affinity to the binding site, but there are 10 of them, so avidity is high. After binding, the strength it produced is high, so avidity is high. So the binding capacity, after the binding, the formation of antigen antibody complex, the strength of the binding is high, the avidity is high. So I hope you understand the difference between affinity and avidity. So let us move ahead. And the immunology basically refers to all the in vivo interaction, whereas serology refers to the in vitro antigen antibody reactions. Therefore, immunology includes the reactions between antigen and antibody like hypersensitivity, autoimmunity, pathology, and so on. Whereas serology include antigen antibody reactions which are used for lab diagnosis of infection, where you detect the antigen or you detect the antibody. So this is basic difference between immunology and serology. Though we are talking about antigen antibody reactions in immunology, then it is inside the body. If same thing in serology, that is in vitro. That is basic difference.